And it's possible that C4 rice would be an example of that. But that's going to be a difficult project. I don't think success is certain, though I'm certainly impressed by the attempt that's being made. But in cases where natural selection has been working not in the direction that we would choose, but counter to human interest, those are the cases where there may be a lot of relatively simple genetic changes that we could make that would improve the crop from our point of view, changes that have arisen repeatedly over the course of evolution but have never become established because they've lowered the individual fitness of plants. And so my next two lectures on Monday, I will be talking about trade-offs, both the kinds of trade-offs that I think constrain past natural selection but still constrain us in terms of our ability to make improvements, things like a limited supply of some resource, including resources like time, perhaps. And then moving on to a more optimistic view for the rest of the series, where I talk about the opportunities that natural selection's limitations have left for crop improvement by humans. So thanks very much for your attention, and if I have time, I'll take questions.
Thanks. Yeah, we have plenty of time for questions, so who would like to start asking a question? Or making a comment in case you
that's fine.
have a major disagreement of some kind.
One of the great things about getting old is I no longer feel stupid when I say something stupid, so go ahead and tell me I'm wrong.
Okay, Paul. Maybe.
Thank you for that very stimulating lecture. I really enjoyed it. Could you comment on the evolution of networks? And I'm thinking about simple mutations allowing complex traits to be induced because of the fact that we have genetic regulatory networks where the effect of one gene can affect many. Is there a selection for networks?
I'm not sure if I can do that question justice. I would say, one of the cases that I criticize in the book is work by Monsanto, where they claim to develop a drought-tolerant corn. And this was a high-profile paper in PNAS. And that was a case where the change that they made was a change in a transcription factor. So there were, many different changes, many different genes that were controlled by that. I don't see that as fundamentally different than a change in amino acid in a protein, because just in terms of how easily it arises, you've got a bunch of bases in the promoter region. Any one of those changing could change the binding to the transcription factor, or a change in the transcription factor itself. Seems like stuff that could arise pretty easily. In terms of the question you really asked about networks, I don't know.
One of the things that's often crossed my mind has been the availability of soil nutrients during evolutionary history, in particular nitrogen. When we're growing crop plants, or when we've been growing crop plants over the last few hundred years, we've been giving fertilizer of various sorts, and that's been given perhaps the opportunity to manifest characteristics that perhaps wouldn't have been dominant in the evolutionary history of that. I wonder, we map CO2 from ice cores and things of that sort. Is there anything down for nitrogen on an evolutionary scale that would give us some insight into those sorts of things?
Boy, that's a great question. of that was a really cool study where they looked at sediment cores from lakes and were able to recover Daphnia eggs from an 800-year sequence, and were actually able to hatch out some of the 800-year eggs as well as the intermediate ones. And in one case, they actually recovered the pathogens of Daphnia as well, and were able to sort of make this timeline of evolutionary arms race between Daphnia and their pathogens. So certainly you could look at you know, there's a lot of old cores that you could get. Now, you know, nitrate is awfully mobile, so that's not going to stay put. And I don't know about, you know, whether the stability of organic nitrogen would be enough that analyzing the organic changes in organic nitrogen fraction in a core like that would be useful. But there is another more evolutionary approach, and this was I know it was in the first draft of my book. It might have got chopped by the editor, but if so, I can find it. What they did was they wanted to map the Earth's temperature, not using the usual isotope ratio from ice core sorts of things, but using an evolutionary signal. And so they used the method of ancestral state reconstruction, where they had this huge number of bacteria, and they would say, okay, so these two had a common ancestor 10 million years ago, and we can infer from their present-day DNA sequence what the DNA sequence of a particular gene was 10 million years ago. And then we can do that for 50 and 100 and so on. And then they made all of those bacteria in the lab. I think they made the bacteria, maybe they just made the enzyme. And they determined the optimum temperature for each of those ancestral enzymes, and made the assumption of, you know, the adaptationist assumption that probably the temperature that the bacteria were exposed to was close to the optimum temperature determined from those resurrected DNA sequences. And in fact, it matched pretty well what we get from the isotope record. So I wonder if we could do something like that, you know, recovering, can we get DNA from pollen grains, you know, recovering cores, getting DNA sequence, and we'd have to know, we're not going to get RNA, right? So we can't do RNA, you know, gene expression. But could we develop some relationship between the DNA or the amino acid sequence of ammonia uptake transporter in roots, for example, reconstitute that, figure out what its optimum ammonia concentration was, and then plot that over time.
I wanted to ask a question which, when I read your book, that came back to my mind, but it has been bothering me for many years, and I'm asking it not being a plant breeder, so I can ask that kind of dumb question. But it relates to your point on individual fitness versus community performance. So does that mean that breeders have got it all wrong? Chasing after an individual gene by trying to phenotype an individual plant, or even selecting an individual plant, as opposed to looking at community performance? What implications does that have for plant breeding as such?
Well, maybe it's my third lecture, I'm going to talk about a really interesting paper that was done here. I'm afraid I don't remember the authors, but they're probably in the audience, looking at breeding for yield versus phenotype approach to breeding. And so I will discuss that issue then. I think it really depends on what the trait is. For something like disease resistance, probably you can look at an individual plant and tell whether it's susceptible to that disease. You might be missing some strange kinds of things. I mean, canopy architecture is going to affect humidity in the canopy, for example, and that might have an effect on disease susceptibility that wouldn't show up in an isolated plant. So I think looking at, I mean, individual genes is fine, but a breeding program that's based just at looking at individual plants for most of the program, and then only quite late in the program doing the yield tests. I think there's a possibility there that plant breeders are passing up some useful traits at early stages, and then in the final yield testing, selecting the best yielding from a population that doesn't necessarily have all of the potential of the original cross.
So we have to get to the yield testing faster.
Well, so that's one approach, and the other approach is, are we smart enough to figure out what traits we should have in an individual plant that will make them work well as a group? And I think Donald did a fairly good job on that.
Near the start, you talked about that aquatic wild rice ecosystem, that it was a dumb question to ask what the function of that simplicity. And yet, these days, a lot of ecologists like to talk about ecosystem function, ecosystem services, things like that. And evolutionary biologists, when they create ecosystem level models, they see some sort of progression from totally chaotic ecosystems to what the mathematicians call self-organized chaos. So something's happening. So my question would be, do you think it's done to ask those questions about ecosystem services and so on? And if not, what's the best way of approaching it? Would it be at looking at what individuals do, or would it be to try and study all ecosystems?
Well, there is, ecosystem services are fine. I have no argument with that. I do make the point that I think the conservation biologists have leaned too heavily on ecosystem services as the main justification for preserving natural biodiversity. And when I made that point at the conference, the CGIAR conference in Beijing a couple of years ago, a couple of people who know a lot more about this than I do came up to me and said, people don't use that as the main argument for preserving natural biodiversity. So apparently I've been traveling in the wrong circles. But this self-organizing, I really don't buy. That is to say, clearly any system that has a bunch of interacting parts, various things are going to happen, and you're going to get certain kinds of feedbacks and possible stability and that sort of thing. So I'm not at all arguing that if you go out to a natural ecosystem, let me rephrase that. If you go out to a natural ecosystem and you were to figure out all of the feedbacks, positive and negative, that are happening there, you would certainly find many feedbacks that do improve ecosystem function in terms of stability or resistance to invasion or whatever. But what I'm arguing is that you wouldn't find that as a universal result, that the kinds of feedbacks that you would find in a natural ecosystem wouldn't necessarily be better than what really smart agricultural ecologists would think of and design, because we haven't had. I made the argument here for natural selection, and I make that argument in the book, that natural selection doesn't improve ecosystem function natural selection can improve ecosystem function because a forest full of disease-resistant trees is more stable. but natural selection doesn't consistently improve things like the number of species, or whether they're clustered together by species or more dispersed, or patterns over time, you know, those kinds of things. The people who know most about this stuff, I think will tell you that I'm right, that that's not an expectation of natural selection. It's not a universal opinion among evolutionary biologists any more than global warming is universal among climatologists, but it's about the same ratio, I think. Something else. Oh, right. The question then is, okay, if natural selection, which we sort of understand, doesn't improve the overall organization of ecosystems, are there other processes perhaps? And I've had a recent debate on my Darwinian Ag blog with somebody from the Land Institute who argued that maybe ecological succession would consistently lead to improvements. And I cited some counterexamples, and he had some arguments and so on. He's a smart person. But in the book, I said, well, this is kind of an empirical question. Let's compare natural and agricultural systems by criteria that are relevant to agriculture. Because, there might be processes other than natural selection and succession. Maybe there are processes that lead to these kinds of improvements. So if so, we would expect natural ecosystems to be more stable, productive, more efficient, and so on, relative to agriculture. And it's really hard to find the right kind of data to make that comparison, because you can't really compare a natural ecosystem with no inputs and with species that have low harvest index because they haven't been optimized for grain production, with a system that has a lot of inputs and that has plants that have been bred to meet our needs. And so the two cases that I found that seemed like the closest to the right comparison, but I bet there's better ones out there. The two cases that I used were wild rice, natural cultivars of wild rice, managed by humans without fertilizer, where I couldn't find a really good data set, but it looks like the human managed systems do a bit better than the natural systems in terms of both average yield and stability over years. And then a somewhat better example looking at rainbow and caribou and limiting the data to cases where the caribou are grazing wild lichens, the reindeer are grazing wild lichens like the wild caribou, rather than cases where they're getting artificial feed. And there again, we see higher productivity of reindeer relative to caribou, and although both human-managed caribou and natural caribou herds have gone through some sort of population crash boom cycles, they're more severe in natural populations than they have been in human-managed populations. So I don't think it's that. The example I took out of the book is the failure of the Biosphere 2 project, which was supposed to self-organize and keep everybody healthy and happy and so on. That got cut by the editor. So I don't think the evidence is totally conclusive, but all the evidence I can find doesn't support the superiority of the organization of natural ecosystems, especially if you're talking about exporting food to cities, which is something natural ecosystems don't typically do.
Yeah. Sometimes I am wondering why most of the cultivated plant species are polyploids, like their wild ancestors. Even rice was also reported as secondary polyploid, cultivated rice. So what is your opinion about that? Just on natural sequence.
So you're saying the plants that were domesticated were already polyploids?
Most of them are polyploids.
Yeah. Interesting. I mean, certainly one thing that humans look for ease of harvesting often is large seed size. And many of our cultivated plants have larger seed size than wild species, which has all sorts of implications. I don't know if that would be effective of polyploidy, but certainly, I don't know, do you have thoughts on that? Okay.
I'll answer your question.
There has been a study.
Yeah, if you ask me hard questions, I'll make you answer.
There has been a study that shows that the cell size increases with deploying.
Okay, and
Fill parameters.
so larger cell size, and
So you get bigger organ size.
okay.
Okay, hey, maybe that's the last question.
Back to the question about natural system and agriculture. I recall that in my graduate school days that Dr. Martin Wolf in England advocating varietal mixtures as a means to control diseases.
Yeah.
And so we know that if you have a mix of genotype in a field, increased ability, in this case disease resistance. But then the whole system probably must fail because our strive for uniformity, for harvesting, and so on. So I wonder if you look in the future, whether that system of selecting for diversity within field may come back. They don't have to be all mixture, they could be patches. Yesterday we have a visitor from Yunnan province, and they talk about how the whole valley planting each village, let's say half a hectare, planting different genotypes. So each village can be a 15 genotype altogether. It's about right. And so I wonder whether as we move forward, like your future scenario, plant breeding may aim for more diverse genotype in a variety rather than a homogeneous single genotype.
Well, I certainly advocate greater diversity, and the question is how that diversity should be deployed in time and space. And so, if I understood you right, we're talking about this valley having 15 different genotypes in the valley, but not 15 genotypes all mixed together in a field. Is that right?
There really is even family, like a co-op. Make a determination that I'm planting this variety, you planting that in patches.
Yeah.
And that's as a means to control, in this case, rice disease.
Yeah. Yeah, so I think that's quite an important question. Clearly, diversity has an important role in controlling the disease. And the kinds of questions that I ask in the book is, let's say we've got 3 crops that we can grow in some region that are, there's a reasonable market demand for, and so on. We could grow them as a mixture of corn, bean, and squash, or we could grow them as a rotation, a 3-year rotation of corn, bean, and squash. And the point I'm going to make in, I think, my last lecture is that people have never done that comparison, comparing the same level of diversity as a mixture versus a rotation. As far as I can tell, there are lots of 1-year comparisons of intercropping with monoculture, but not the 6-year comparison that you would need to compare a 3-year rotation with a 3-crop mixture. And I will talk about that and show the results of some simulations by Dr. Sue, I forget. It's in the book. I will be showing that in one of my lectures, addressing the relative merits of intercropping versus crop rotation over multiple years.
Good. I think we'll have to stop it here. We're almost at 2:30. But that's
Thank you for staying.
a very interesting question to come back to next week. So Monday, 9 o'clock is our next topic. I hope that we can get the average age of the people asking questions below 55 then. Don't be shy. So let's give the board a big hand and see if I can